Last time we talked about the differences between DNA and RNA. We touched briefly on transcription. Today we're going to dive deep into transcription and translation, talking about the various bases that RNA and DNA have. So let's get started. Now, the first step in decoding genetics is to transcribe from DNA to RNA. And we talked last time about how the mRNA makes a copy of the DNA, the mRNA being a blueprint of the master copy. And we called that transcription, which is when the RNA is going to transcribe from the DNA. Now, the transcribed code is used to make proteins. Long chains of amino acids which form proteins are called polypeptides. And you can find as many as 20 different amino acids in polypeptides. The specific amino acid in a polypeptide and the order in which they are joined determine the properties of the proteins being formed. The sequence they are in also helps determine the shape, which determines the function. RNA contains 4 different bases. Contains adenine, just like DNA does. Contains cytosine, just like DNA does. Contains guanine, just like DNA does. Contains uracil, unlike DNA does. So DNA and RNA share these, when we're talking about the bases, shares these 3, adenine, cytosine, guanine. DNA does not contain uracil, only RNA. And the body knows whether it's going to be DNA or RNA because of this last base here, which is called uracil. Now, these bases form a language using 4 letters, A, C, G, and U. It's much shorter to use letters instead of writing out adenine, cytosine, guanine, or uracil multiple times. So they just use letters because each one starts with a different letter, so it's easy just to abbreviate. Now, this language is called the genetic code. Well, how can a code with 4 letters carry instructions for 20 amino acids? Well, the genetic code is read 3 letters at a time. This means each word is 3 letters long and corresponds to a specific amino acid. These 3-letter words in the mRNA are called codons. Now, a codon consists of 3 consecutive bases that specify a single amino acid to be added to the polypeptide chain, which is a long chain of amino acids. Now, if you're wondering, how does the body know how to read that word? We're going to talk about that momentarily. So how do you read a codon? Well, there are 4 different bases, A, C, G, and U, which means there are 64 possible 3-base codon combinations in the genetic code. Now, most amino acids can be specified by more than one codon. Here's an example. Here we have UUA, UUG, CUU, CUC, CUA, and CUG. That is for the amino acid leucine. Now, sometimes there's only one codon that can specify an amino acid, like UGG. That's the amino acid tryptophan. Now, there is a genetic code table that makes decoding codons super easy. In the next slide, we're going to show you how easy it is. All right, so let's take a look at this. So you're always going to start from the center and work your way out. And the example we have here is UGG. We had that on the last slide. And you can use this for any 3 letters you have as long as it's ready to be translated. So we have U, we have G, we have A, and C. So we start in the center with U. So this is my starting point. Now that I have my starting point, I am working within this grid right here. I'm not working in these other 3/4 of the grid. I'm working just right here with 1/4 of the entire circle. My next letter is G, and I can choose U, C, A, or G because those are the only options. I choose G, and that has narrowed it down to this part right here. And my last letter is G. So when I have UGG, that tells me right here it's tryptophan. It is that easy. So you start in the center, and you just work your way out. Every language has punctuation marks that tells you when to start and when to stop. Well, the genetic code does too. The methionine codon, AUG, serves as the initiation or starting point for protein synthesis. Following the start codon, mRNA is read 3 bases at a time until it reaches one of 3 different stop codons, which ends translation. Once it has ended, the polypeptide chain is complete. All right, translation. Well, what role does the ribosome play in assembling proteins? We said ribosomes are the site of protein synthesis. They make proteins. Well, what does it actually do in assembling the proteins? Once the polypeptide is complete, it folds into its final shape or joins other polypeptides to form a functional protein. Putting a complex toy together requires both the instructions and putting it together piece by piece. You don't go from step 1 right to step 10 right away. You have to follow it in order. Step 1, step 2, and so on. Ribosomes carry out reading the instructions and putting the parts together. They use a sequence of codons and mRNA to assemble amino acids in the polypeptide chains. The coding of mRNA message into a protein is a process called translation. Now, transcription happens before translation. Remember this and don't confuse the two. So mRNA makes a copy of the DNA. That is transcription. What they do is they decode that mRNA by tRNA, and that is translation. So transcription happens, mRNA makes a copy of DNA. tRNA comes in and will translate the mRNA message into a protein, and that is translation. So there are steps in translation. Transcription isn't part of the translation process, but it is critical to it. So they're two separate processes. Now, transcribed mRNA directs that process, and that is the transcription. In a eukaryotic cell, transcription goes on in the cell's nucleus, and translation is carried out by ribosomes after the transcribed mRNA enters the cytoplasm. So after the mRNA leaves the nucleus and goes to the cytoplasm, that's where translation is going to be carried out by the ribosomes. Now, when the ribosomes attach to the mRNA molecule, translation begins. So the first step, when a ribosome attaches to an mRNA molecule in the cytoplasm, translation begins. We just said that, and that's the very first step. As each codon passes through the ribosome, tRNAs bring the proper amino acids into the ribosome and attaches them one at a time to the growing chain. Each tRNA molecule carries one amino acid at a time and has 3 paired bases called an anti-codon, and these are complementary to one mRNA codon. Well, what does that even mean? Well, there are complementary bases. A pairs with U, and U pairs with A. C pairs with G, and G pairs with C. And we'll talk about what that means a little later on. So step B. The ribosome helps form a peptide bond between the first and second amino acid. At the same time, the bond holding the first tRNA molecule is broken, and the tRNA moves into a third binding site, exiting the ribosome. The ribosome moves to the third codon, where tRNA brings in the amino acid specified by the third codon. So if the codon specifies AGU, then the anti-codon, the tRNA bases that complement it, will bring UCA. So just a reminder, A pairs with U pairs with A, G pairs with C, and C pairs with G. So here we have mRNA AGU. That's from the mRNA. The tRNA is going to complement that. Well, what pairs up with A? That's going to be U. What pairs up with G? That's going to be C. What pairs up with U? That's going to be A. So you can think of these as puzzle pieces. They're going to fit together perfectly to carry out the amino acid and protein that it is building. So the polypeptide chain continues to grow until it reaches a stop codon on the mRNA molecule. When it reaches a stop codon, it releases both the newly formed polypeptide and the mRNA molecule. This completes the entire process of translation. Now with that, we're almost done. So the roles of rRNA and tRNA in translation. Now all 3 major forms of RNA, remember we talked about the mRNA, the rRNA, and the tRNA, they come together in the ribosome during translation. mRNA directs the entire process. The tRNA delivers the right amino acids called for by the mRNA. Think of the tRNA molecules as adapters that enable the ribosome to read the mRNA's message accurately and get the translation just right. Ribosomes are composed of roughly 80 different proteins and 3 or 4 different rRNA molecules. These rRNA molecules hold ribosomal proteins in place and help locate the beginning of the mRNA message. They may even carry out the chemical reactions that join amino acids together. So you can see that rRNA, tRNA, and mRNA, they all work together to make these chemical reactions happen that join amino acids together to build these proteins. So the molecular basis of heredity. Now proteins have everything to do with traits. Your gender, your eye color, your skin color. Any trait that you have, it's because of the protein. Now, many proteins are enzymes which catalyze and regulate chemical reactions. Genes can code for enzymes. One that produces pigment can control the color of a flower. A gene that produces proteins can regulate tissue growth. Proteins are microscopic tools where each is specifically designed to build or operate a component of a living cell. Once scientists found genes were made of DNA, molecular biology became established. Molecular biology seeks to explain living organisms by studying them at the molecular level. All right, so last one. One of the earliest findings became known jokingly as the field's central dogma. Now, the central dogma of molecular biology is that information is transferred from DNA to RNA to protein. Gene expression shows the way in which DNA, RNA, and proteins are involved in putting genetic information into action by cells. The genetic code in almost all organisms is universal. If you look at any organism, you're going to see adenine, which is the A. You're going to see cytosine, which is the C, guanine, which is the G, and uracil, which is the U. It's found in almost every single organism. And this is what we mean by the genetic code being universal. The code is read 3 bases at a time in every organism, and it's always read in the same direction, left to right. It never changes. It will always be read 3 bases at a time. It will always be read left to right. Now, despite enormous variety in species, living organisms display remarkable unity at life's most basic level, the molecular biology of the gene. And with that, we're all done. If you have any questions, post a comment below, and we'll see you guys next time.